We welcome you, Ryan, and we're excited to hear all the work you're doing. In addition, I would like to grant you a pin because you are a scientific hero to FAS.
Thank you. Pointer. Okay, thank you. Hello, good afternoon. Thank you all for inviting me here today. This is my first in-person conference since before COVID. I had dust all over my dress shoes. It was crazy. Also, I want to give a shout out to my daughter who just turned 9 as well. She might be watching me. Love you, Tiger. Today I'm going to be talking to you about RNA gene therapy for Angelman syndrome. Most of this talk, and I didn't know this beforehand, but most of my talk has been described already, but very eloquently and very beautifully by other speakers, so this may be relatively short. If there's one thing I want you to take away from this is that if this approach works, then it's the stop approach, and it could, in theory, be a one-time injection that should last for the life of the patient. So just a disclosure before I begin. This work is funded by Taysha Gene Therapies Incorporated, and I do have a personal financial interest in Taysha. UT Southwestern also has a financial interest in Taysha through exclusive licensure. So this was already described by Allison, but when neurons communicate with each other and when they're developing, they're constantly producing new proteins. And so these proteins are very important to develop, but it's also important that they are degraded after they've been used, or to be recycled or to be flushed out of the body some way or another. And so that's really what UB3A is doing. It's the degradation of proteins which are no longer needed. So it tags a substrate for proteasome degradation. And now, how this relates to Angelman syndrome. So UB3A, the lack of UB3A can either directly or indirectly lead to the phenotypes associated with Angelman syndrome. And it could also lead to the mosaicism, which was described earlier. All right, so when we talk about neuronal maturation, what we're really talking about is the development of new connections between neurons and the ability of those connections to actually transmit neurotransmitters, that sort of thing. And so when you have a buildup of these proteins, and two in particular, ARC and FXN5, that actually is inhibited. You no longer have this dendritic growth, and you no longer have the ability for the neurotransmitters to be received by another neuron. I won't go much into this, but as we've already described previously, the maternal allele for UB3A is active, whereas the paternal allele is silenced. Now, with the stop approach, in theory, this could treat all of these different genotypes of Angelman syndrome. There's a question about whether there'll be too much for UPD. There's also a possibility that the paternal allele could also have a mutation. There's no evidence that I'm aware of how frequent that is, but if there's a mutation or dysfunction in the paternal allele, then this stop approach would not work. And again, like I said, I prepared this. I didn't know that everybody else was going to talk about it already, but the paternal silencing is mediated through a long non-coding antisense transcript, and a portion of that is UB3A ATS. Now, what's really what's really a great aspect of this UB3A ATS is there's really no functional role for it other than to inhibit UB3A. So from a therapeutic perspective, inhibiting that is really cool. So basically, on the paternal allele, you have the Prader-Willi syndrome imprinting center, and on the paternal allele, it is not bound, and therefore on the paternal allele, you have a transcription of the entire long non-coding RNA. Now, on the maternal allele, you do have methylation, and so therefore the maternal allele is that long non-coding antisense transcript, including UB3A ATS, is not transcribed. And then with the Angelman syndrome, of course, on the maternal allele, it's not functional or it is deleted. And so our approach here, very similar to the Ultragenyx approach, the ASO, and other stop-to-stop approaches. And so this all started from the Meng et al. Paper from the Baudet lab in 2015, where they showed that an antisense oligonucleotide targeting the region between SNORD115 and UBE3A was effective in alleviating the Angelman syndrome phenotype in the mouse model. Further studies later on also described how incorporating SNORD115 into that therapeutic target region could also alleviate the phenotype in the mouse model. And so our approach would be to use a short hairpin RNA to target this region and vectorize it. So, already described that, but so there are complications with using siRNA or shRNA, and I'll describe them as best that I know. But one thing that in terms of the region to which we are targeting now, we want to be sure that our short hairpin RNA does not lead to degradation of the genes right next to it, so that are critical for Prader-Willi syndrome. And so the last thing we would want is to cure Angelman syndrome and replace it with another equally devastating condition, Prader-Willi syndrome. Okay, so there's been some talk today about adeno-associated viruses. And so just to recapitulate that, there was a clear line in the sand when it comes to gene therapy and its use and the ability to use it. So there's before AAV9 and there's after AAV9. Before AAV9, it was definitely a sexy topic. You could talk about it as much as you want and about how it could cure all the diseases, but it wasn't until AAV9 came along that it actually could be used therapeutically. So the pros for adeno-associated virus, wild-type AAV does not cause any human disease by itself. It's got a low immune response compared to other viruses. Recombinant AAV9 or AAV does not integrate into the human genome, and it can target a wide variety of tissues and organs. So some of the cons, and it's not really related to this talk, but it's got a fairly small packaging size, 4.6 kb for single-stranded AAV9, and I'll talk a little bit about that, and 2.3 for self-complementary AAV9. That being said, for short hairpin RNA, it's roughly 50 base pairs in total, and then you have promoters and everything, but it fits well within the self-complementary AAV9. And so just a little bit about self-complementary AAV. This was shown in the early 90s, I believe, that if you fold the plasmid in on itself, it's called self-complementary, you can increase the efficacy of transduction 10 to 100-fold. And so anything below 2.3 kb is ideal for this. And like I said, there was actually a concern that it was too small. We had to do some pilot experiments to make sure that we could actually manufacture viruses which have a construct so small, and it worked fine. But just so you know, that this is not an issue for this particular project. Okay, so when we're talking about potentially injecting a human being with an shRNA, we have to be very careful about certain things. And so the first thing we want to investigate is off-target effects. As few as 6 base pair complementary could lead to off-target binding. It's a very small number. It wouldn't be to the same extent as like the 21 nucleotides, which is what shRNA is, but there could be some effect. And so the first thing we did was a bioinformatics analysis to get all the potential candidates which could in theory bind to this therapeutic target region. And we came up with over 2,000. And that's really important because by the end we did not have 2,000. So then we take those 2,000 candidates and we do further bioinformatics analysis to determine how likely they are to bind to other sites. And if they did not meet a certain threshold, we did not include it in our as a final candidate. And then step 3 is to check it against the monkey background, because no matter what, we are going to have to do, if it looks promising, we're going to have to do a safety test in non-human primates. And so in order to be effective and actually to be relevant, it has to bind to the monkey genome, and it also we don't want to have off-target effects in the monkey genome. So a lot of what we're doing it's very similar with the human cells and the mouse cells, and then we go on to mouse behavior. But Dr. Yonghui Zhang earlier talked about the induced pluripotent stem cells, and I believe that my lab was the first recipients of them. So if you're out there, Dr. Yonghui Zhang, thank you very much. We are using them, and they are working. So we had these induced pluripotent stem cells. They already did that part, which is quite expensive and time-consuming, but they sent it to us as iPSCs, and in my lab we have basically differentiated them into neurons. And so we do have healthy and we do have Angelman syndrome iPSCs. We have it for the UBE3A deletion and the UPD genotypes. Now, very similarly, in the mouse model, there's a reporter line which has a fluorescent tag on the paternal UBE3A. And so if this stop-to-stop approach works, then what you see are glowing yellow neurons. And so similarly with the human iPSC-derived neurons, if there's UBE3A, you can pretty much know that it's from the paternal side. For the mouse, if it's glowing yellow, it's paternal UBE3A. So, okay, so like I said, we test these candidate siRNAs. So just to back up a second, I talked about having 2,000 candidates. This is actually siRNA, and these are oligonucleotides. And so we tested these siRNAs on both the human cells and then had mouse-specific candidates on the mouse cells. And then we are basically measuring for the common UBE3A targets like UBE3A, UBE3A ATS, and other potential off-target effects and the Prader-Willi syndrome critical genes, things like that. Go on to this. Now for the behavior for the mouse in vivo studies, basically what we did is we took our siRNAs and converted it to an shRNA and packaged it in an AAV9 and delivered this intrathecally to a mouse which is 7 to 10 days old. Intrathecal means into the spinal cord, by the way. And then we wait 8 weeks and we do the first behavior battery tests. These are very, very commonly used behavior batteries, including fear conditioning, rotarod, things like that, just to test for cognition, balance, things like that in the mouse. And I was actually very pleasantly surprised yesterday to see how ubiquitous this behavior paradigm is across labs. And then after the first behavior battery, and that all those tests are done on a single mouse, we repeat the exact same behavior battery 4 weeks later. And then, like I said, we measure for UBE3A, UBE3A ATS, and other things. So in addition to off-target binding, another thing to consider, and if this ever gets to the clinic, it's definitely something parents should talk about with their doctor and things like that. But with shRNA, it has been shown that vectorizing it can lead to cardiotoxicity through endogenous microRNA competition. And basically, it interferes with the processing of endogenous microRNA. Now, since that study has come out, which showed that, there have been follow-up studies which have showed that can be mitigated by creating a microRNA scaffold around the short hairpin RNA. And so when you inject that type of plasmid or virus into a mouse, it alleviates the cardiotoxicity without reducing efficacy. And so a year from now, I hope to be back here and I will have a much more robust data set. Admittedly, this is probably the only data slide that I have today. But what this is a screen on neuroblastoma cells. So a cancer, neuroblastoma cells from a human. And so this is just the test of the 46 final candidates that we had, the siRNA candidates. So like I said, we had started off with 2,000. The bioinformatics analysis whittled it down to 46 based on off-target binding. And then we tested that in vitro on this neuroblastoma cell line. And so, I mean, it's not a healthy cell line. It is a cancer line, but it does have healthy UBE3A levels already. So now as I describe this data, bear that in mind. So what we're looking at with the blue bars here is that each one of these final 7 candidates, which we have, there's a significant reduction in UB3A ATS. And so that's really important for obvious reasons. Now, in terms of the increase in UB3A, that's sort of modest or non-existent in these cells. We'll see what that means later on, but we're not as concerned about that at the moment as we are the reduction in UB3A ATS, because among other things, it already has UB3A. And the second thing is we don't know the kinetics well enough at the moment to say how long it takes after inhibition of UB3A ATS for there to be an increase in transcript of UB3A. But these are our final 7 candidates, which we are going to be vectorizing and testing in the human iPSCs, derived neurons. And so this is a picture from the mouse cerebellum. And so what we're showing with the top left panel there, the green, is that the virus did reach the cerebellum after an intrathecal injection. And what we're seeing is it's modest, but it looks like a slight improvement in the bottom left panel. That's UB3A. And so in the Purkinje cells of the cerebellum, very important region for Angelman syndrome, it does appear to be that there is an increase in UB3A. But this is pilot data at the moment. Like I said, a year from now, I hope this will be much more robust. And so I don't know how many parents were at my talk about 7 or 8 months ago, where I was trying to recruit you to donate your fibroblasts from your children, and I appreciate that very much. That was actually, we knew that Dr. Yanghui Zhang was producing iPSCs, but we had no idea when they'd be ready with the COVID shutdowns and things like that. And so we anticipated using your iPSCs and making our own iPSCs. It turned out by the time those were ready, Dr. Yanghui Zhang already had them, the iPSCs ready. So your children's fibroblasts are at Coriell right now, and I'm sure that they will be used in the future by other laboratories. But I just wanted to let you know that I have received communication about that in the past. So that's where they are, if you're wondering. And so that was what I talked about. And the important thing about iPSCs, for that therapeutic target region, which I talked about earlier, it is not well conserved between mouse and human. And so whatever siRNA, shRNA we find that works in the mouse, that will not work in human. We have to redo it completely in the human patient cell line, the iPSC. So that's really why it's important to get this. Move on. Too many slides. Okay, now when we have the IPs, this is just preliminary data showing, or experiments showing that what we're doing is working with these iPSCs. So we have the iPSCs, and then the stages go from iPSC to neural stem cells, neural progenitor cells, neural precursor cells, and neurons. And at each step of the way, we actually make sure that it reaches that stage with the cocktail of reagents that we give to it. And so it worked. We had the iPSCs, and this marker showed that they were iPSCs. And then same thing with neural stem cells and neural progenitor cells. And I'll just point out that at this stage, the healthy control and the UB3A loss look very similar. Moving on, once we get to the neural precursor cells, and especially with the mature neurons, we actually can see in a dish what's happening with Angelman syndrome. And so we see the maturation of these neurons is dramatically reduced in the Angelman and the UB3A loss compared to the healthy neurons. And so we're using that just to know when we should actually incubate these iPSC-derived neurons with our plasmids, with our siRNAs, that sort of thing. And so that's where we stand with that right now. And we are also measuring UB3A and UB3A ATS throughout those experiments. And what we're finding is that UB3A ATS does not come along until the neural precursor stage. And so just bear that in mind. All right, let's skip off. Okay, and this is just some pretty pictures which show that our plasmids, and so that is what we actually will be using to prepare the viruses. And so it's a circular DNA construct, which are put into the AAV knives. And these constructs have GFP. And so these are on the iPSC-derived neurons. And one thing in terms of troubleshooting, making sure everything works, we have to make sure that the plasmids actually go inside the cell and express. And so they are expressing GFP, meaning they are working. and I'm way over time, so I'm just going to skip on to this and say that hopefully in a year from now we'll have all this shRNA candidates, and they'll be packaged into AAV9, tested on iPSCs, and hopefully we'll be close to starting NHP safety assays and that sort of thing. And I just want to acknowledge, so I deserve no acknowledgement beyond the fact that I can hire really good people, and that starts with the first two ones on here, and that's Hairi Kang and Violetta Zerig. It's hard to really convey how difficult it is what they're doing. And I don't want to have to come to this talk and say that, this treatment isn't ready because we forgot to do something one day. They're really talented scientists. So thanks to Hairi and Violetta and also everybody else on my team. Thank you to Dr. Youngwei Zhang for the iPSCs and also other members at UT Southwestern, and Taysha, of course, for funding everything. That's all I have. Thank you very much.